So today what we're going to do is try and pick up where we left off. Be a bit of a challenge to do so. And where we left off last time was using Bedtools for some real-world analyses. So that's kind of where I'm starting here. And I apologize, this is super odd for me to teach this way, not seeing anyone, so I'm going to struggle. I'll do my best though. I'm going to skip ahead to slide 32, which is where we left off. And the motivation behind this question is basically, a lot of times we use tools like Bedtools or other methods to find whether or not sets of intervals like exons or enhancers in this case, or introns or ChIP-seq peaks intersect with one another. But that intersection alone tells you that there is some coincidence between those genomic features, but it doesn't tell you whether or not it's statistically significant. And more, what I mean by that is, could that same level of intersection occur by chance, just because of the spatial disbursement of, say, exons and enhancers? So if you got 10 intersections, maybe that would happen no matter what. And it has nothing to say that that degree of intersection has any biological or statistical significance. So there are a number of ways to address this. This is a problem really in all science, trying to convince ourselves that the signal we think we're seeing is real and not some noise. So we're going to walk through an example of how we do that today, using something called a Monte Carlo simulation. And there's a tool in Bedtools for doing this. And I want to just try to spend some time talking about what a Monte Carlo simulation is. You probably have some guess based upon the name itself, and many of you may have done this before. All right, so the question that we're getting at here is whether or not the SNPs that are in that gwas.bed file that you've been looking at probably for homework and we looked at last class, whether or not those SNPs overlap enhancers in these human embryonic stem cells more often than would be expected by chance. And the biological motivation for looking at this question is, I touched upon it last time, but genome-wide association studies, by and large, have revealed single nucleotide polymorphisms in the human genome that are significantly associated with different disease traits like Crohn's disease or macular degeneration or type 1 diabetes. But the vast majority of those SNPs that are strongly associated with those phenotypes are not actually in DNA sequence that encodes proteins. So a lot of the open question has been, well, gosh, what if they're strongly associated but they're not in protein coding genes, what's going on? And one of the early hypotheses was that, well, these SNPs that are associated with complex diseases like Crohn's disease and other autoimmune disorders, they're not Mendelian. There's not one gene that causes them. Probably what's going on is these SNPs modulate the expression of genes involved in pathways that lead to these different phenotypes. So a paper from the ENCODE study a few years ago basically showed that when they identified enhancers in various cell types and tissue types, that these enhancers coincided with statistically significantly associated SNPs from GWAS studies, suggesting that these enhancers are actually, these SNPs lie in enhancers that are regulating the expression of genes that are connected to these different disease phenotypes. So that was kind of a hallmark finding in the big paper from ENCODE a number of years ago. So we're going to try and reproduce that result, essentially. So we've got our set of SNPs from these GWAS studies, and we've got a set of enhancers from human embryonic stem cells. We learned how to grep that bed file for different enhancers. So we've got our two sets of bed interval files that we want to compare. So we've got the four pieces of information we need are the two files, which we just walked through. We need to measure the number of observed overlaps between the two. And I think, as you probably know by now, that's just using Bedtools intersect. And then lastly, we need to compare the observed number of overlaps to the number of overlaps that we expect. And that observed versus expected ratio is essentially the effect size that allows us to assess the significance of the relationship between the two genomic features. So as a straw man, if we calculated that 100 of these GWAS associated SNPs overlap enhancers, that's what we observe. But then we go on to find that, well, actually, any random set of SNPs or any random set of enhancers are going to overlap at about 100 overlaps. That tells us that what we observed is actually not all that interesting. It's exactly what we'd expect by chance, so there's really nothing to publish there. If, however, that observed number is way higher, or potentially way less than what we'd expect by chance, then it's at the tails of some distribution of what we might expect, and therefore potentially more interesting. It's more of an outlier observation. So obviously what we're going to do here is, this is sort of recapping how we went about grepping for enhancers. So we're including both weak and strong enhancers because we're just grepping for the word enhancer here. We're fixing up the chromosome labeling issue that sort of plagues genomics day in, day out. Everything's going to start with we're going to get rid of the CHR label with this find replace syntax in this stream editor tool called SED, which we learned about. And we could also embellish this by getting building upon the bed file that we actually have and get one that actually lists phenotypes. So this is another file that we could download, and instead of just having the chromosome start and end position as well as the ID for each SNP, we can get a PubMed ID associated with the paper that describe the association, in this case, between this SNP and whether you're a morning or a night person, or this one, which is correlated with BMI and other things. So if you want to use that, you can. So in total, there are 39,335 SNPs like that in the GWAS file, and there are 240,000 enhancers from human embryonic stem cells. And when we intersect those and get a unique, we use this unique option such that we're only counting GWAS SNPs once. So if they overlap multiple enhancers, then we just want to count that overlap once rather than having it report each SNP multiple times if it overlaps enhancers, because we're basically just trying to get a set of the fraction of GWAS SNPs that actually overlap enhancers. So in this case, 2,040 out of the 39,335 SNPs overlap at least one predicted enhancer. So 5.2%. So the question now is, that seems kind of cool, 5%. Is that interesting? How would we actually go about knowing that? Before I move on, any questions at this point? Any technical difficulties? Everyone good? Kind of flying blind here. All right, so I'm going to assume that's a yes. If you have any questions, chime in. There's also a chat option. If you want to raise questions along the way, I'll try and keep an eye on that as well. Okay, So the idea about how we derive an expectation comes from this thing called a Monte Carlo simulation that I talked about. And the Monte Carlo aspect comes from the fact that, as you probably know, Monte Carlo is a big casino place in the south of France. And it gets at the notion that to develop an expectation, we really need to assess how likely the observation that we made is by chance. And in genomics, the standard way to do a Monte Carlo simulation to derive this null hypothesis, the expectation, is to randomize or shuffle the locations of the intervals many thousands of times. And each time we shuffle the location of the intervals, we ask what the intersection is for each iteration of the shuffling. So concretely, what we would do is we would take all the GWAS SNPs and all the enhancers. They have their stated locations in the Bedfile. We could use a tool to randomly replace them on chromosomes. They have new locations now. We take those new locations, we measure how many intersections there are with those new locations, and we ask, well, how many intersections were there? Is it basically 2040 as we observed, or is it far less or far more? We repeat that experiment many, many times, and for each experiment, we track the number of intersections that we make when we reshuffle. And that, if we collect all the intersections from each of those reshufflings, say 1,000 times, we can build up a histogram of the number of intersections that we observe from each of those 1,000 Monte Carlo trials. And then we can use that distribution to establish the null hypothesis. That is what we expect by chance. So we have a null distribution, and then we can ask, well, does our observation lie within that distribution? If so, probably not interesting. And if it's way outside of that distribution, perhaps it is interesting. Okay? Right. So I've got a slide that simulates this that I didn't make. Awesome postdoc in my lab from a couple years ago named Ryan did this. Okay, so let's see if I can get this to work. In yellow are going to be our GWAS SNPs, and in blue are going to be our blue enhancers, or enhancers in blue, sorry. So in this case, what we're doing is shuffling the GWAS SNPs, and then the white lines are the number of intersections observed when we shuffle. And I'm going to redo that. Let's see if I can get it to run again. Painful. Sorry, guys. Yeah, it won't run again. But what you end up with is, for each iteration of that, these white lines are tracked. The number of intersections are tracked over here in this histogram. So what we see is that on average, it looks like the mean of this distribution is like 4.2 intersections when we shuffle things. That way we can ask whether or not, maybe our observation is way out here at 12. So we observe 12, and we can see that that's much higher than what we expect by chance. But if we observed 5, that's right within the expectation, and therefore it's not an interesting observation. Hopefully that makes sense. I can't see your faces to gauge whether I'm talking nonsense or not. If it doesn't make any sense, please chime in and I will try to explain it more. Okay. Okay, could you? I can barely hear you. Can you hear me now? Yes, but very faintly. Right. Yeah, so you can, that's a great question. The answer should be yes. There's a lot of subtlety about how you do these. So in this case, this was really to do things in a simple way for the animation, where you keep one set of intervals in their fixed location and you shuffle the others. It should bear out that the null expectation should come out roughly the same if you shuffle both, you shuffle one and not the other, or you shuffle or you flip it and you shuffle the other one and keep the other one constant. There's some caveats to that though that I'm going to talk about in just a second, but as a basic rule of thumb, it should work out the same way. Any other questions? Okay. Let's see if I can make this work. So what I did here, we're not going to run this. You can run this on your own if you'd like. I want to walk through what we're doing though. So that distribution that I made on the previous slide, I think did 100 simulations, 100 shuffles. So in this case, what I'm showing is how to run this command Bedtools shuffle, which takes these the GWAS SNP locations. It needs to have this genome file, which defines the length of each chromosome, and that's so it knows the boundaries of which it can shuffle the intervals. And that shuffling is the connection to Monte Carlo. You play blackjack in Monte Carlo or in a casino. There's randomization of the cards. That's that, you know, whether or not you bust or hit 21, it should be roughly random. The genome file helps define the boundaries of the deck. The deck here is the genome. Where can we replace these GWAS SNPs within the genome? So this loop is written in Bash, and it says it's a for loop. So for a variable I in the range 1 to 100, that's what the seek 1 to 100. If you type that into Bash on your command line, it'll print out 1 followed by 2 followed by 3 all the way to 100. So we're basically, this allows us to run the command here, this Bedtools command, 100 times. Okay? And what we're doing in each of those 100 experiments is we shuffle the locations of these GWAS SNPs. We pipe the output of those randomized locations of the GWAS SNPs into Bedtools intersect. And now the A file is coming, this dash means that it's coming from as input from the output of the previous command. So the output of the shuffle command is coming in as input for the A file in this intersection. So we're intersecting essentially the randomized locations of these GWAS SNPs with the static positions of the enhancers, just as what was shown as in the animation in the slide before. Again, we're using this -u command to count unique intersections, and then we pipe that to wc-l, and then what we do is we're just keeping keeping track of the count of intersections for each of these 100 experiments in a file called random.intersections.txt. So that will, at the end of the day, that random.intersections.txt will have 100 numbers in it, which are the count of intersections from each of these Monte Carlo experiments. Good? Okay, so, yes?
There was a question in the chat earlier.
Oh, I had to hide that because I couldn't see it. So when I said I was looking at it, I lied. Let's see, let me see if I can find it.
You can hit Alt-H.
Alt-H. Yeah, okay, well, you can. I cannot seem to. Let's see here. Chat. Good Lord. Ah, I see it. Awesome. What test could you use after this to test for significance of whether the intersection is different enough to be significant? Ah, okay. So So basically, how do you know what the p-value is? Is that the question? Yeah, okay. So let me just try and walk through this intuitively. So we've got this distribution, and let's say our observation is out here. I understand the question to be, what test can you use to apply this, to test the significance of this? There's a number of things that you can do. The first is what's called establishing an empirical p-value. So if you do 1,000 trials and the number of intersections that you observe, let me say that backwards. Let's say you do 1,000 trials, and for each of those 1,000 trials, you never observe intersections that are greater than or equal to what you observe. If that is the case, your empirical p-value is p less than or equal to 1 in 1,000. You don't know what the exact p-value is, but you've done 1,000 trials, so empirically, you know that you never saw with the randomization experiment anything that met what you observed. So that's an empirical p-value. The other thing you can do is if this distribution is roughly normal, like a Gaussian distribution, you can use the properties of a normal distribution to ask how many standard deviations your observation is from the mean of that distribution, and once you calculate the mean and standard deviation. So there's standard methods for doing that. I mean, you can calculate that directly once you know the mean and standard deviation of a normal distribution. And I may have time at the end of the course to get into this, but if you're interested more in that, there are some lectures on the SLOBS, SLLOBS, the Salt Lake Learners of Biostats group that I ran last year. There's a couple of lectures on calculating p-values from a normal distribution. And then the last thing that you can do is calculate an effect size. So you observe 12, and the mean is, say, 4. Observed divided by expected, observed over expected, is 12 divided by 4. So the effect size is 3. There's like a threefold enrichment. That's not really a p-value, but it tells you about the magnitude of the effect. Because you could have a very significant p-value out here, but the effect size actually isn't all that big, right? It's only 3 times what you'd expect by chance. Whereas if my laser pointer went way off the screen and what we observed is 1,000, that's going to be very significant, and the the effect size is going to be massive, because then it's 1,000 divided by 4, so your effect size observed over expected is like 250. Let me go back to the chat, because I think the person that... thank you. Okay, that makes sense. Good. I'll use chat. Okay, so moving on. So actually, that's what I was about. The empirical p-value was what I was about to say here. When I do this experiment, when I shuffle the locations of the SNPs 100 times, every value in this random intersections file is 0. There's never an intersection. So that means that my observation of, what did we get, 2040 is definitely, with some caveats that I'll say in a second, is definitely / potentially super interesting, because we observed 2040. When we do this expectation experiment using Monte Carlo, we never see anything greater than 0. So our p-value right now with this experiment is p less than or equal to 1 over 100, 0.01. I bet you if we ran this same experiment 1,000,000 times, we'd probably see 0 intersections every time. If we did that, our empirical p-value would be p less than 0.01. well, whatever 1 over a million is, 0.000001, right? Okay. The problem with this is Monte Carlo simulations make a fundamental assumption that is often violated in genomics. And that is that every nucleotide or every position is independent of one another. So let me say that again. The way to think about a Monte Carlo simulation is that each of the shuffles and the cards that you draw, in the case of blackjack, are totally independent of the previous card and of the next card. And you know, if you've got a good dealer and they shuffle right and they're not cheating you, that should be the case. In genomics, that's a little bit more complicated because there isn't always this notion of independence. Like there's structure to the genome. There are parts of the genome one nucleotide in a gene is not independent from the next nucleotide in the gene. They operate as a unit together. So we tend to violate that notion of independence, essentially, that is central to Monte Carlo simulations. The other thing that's often violated is when I shuffled in this experiment, I'm shuffling to any nucleotide in the entire genome. So this genome file lists every chromosome and then the length of every chromosome. But we've learned 100 different ways so far that there's gaps in the genome. There's parts of the genome that are unassembled. There's centromeres. There's telomeres. I think you could imagine that it's probably not fair to shuffle GWAS SNPs anywhere in the genome. One reason that that's not fair, let me check chat again here. One reason that may not be fair is that we cannot detect a GWAS SNP. We can't even study a GWAS SNP in centromeres or telomeres because we can't assay them. We can't. Sorry, is there a question? Sorry, I can't hear you. I can hear you. I can hear words being said, but I can't make them out. I'm going to try and figure out what's going on with volume. Maybe, can you crank up your audio, your input, or type it into chat? I don't know what its echo mean. Does that mean? Right. Yes. Good point, Travis. I have no earthly idea how to do that. I think you can mute yourself. So volume. Okay, great. Yep, you can mute yourself. So how the heck do I make it so I can hear you? Because this is kind of worthless unless I can hear you. Any Zoom gurus out there that know how to crank up the volume? Turn it to 11. Let's see. Ah, okay. Thank you, Marshall. Can the person that was making that comment about the audio being reverb, can you ask that question again? And I'm going to play with audio, or just someone ask a question, or say something, sing a song, do a little dance.
Hello.
Okay. Got it. It's me, guys. I can hear you guys much better now. I had the volume cranked up on my laptop to max, but within Zoom there's a separate volume. Okay, I think I'm in better shape now. So there were no questions, right? And we got everyone muted. There's no feedback now. Great. All right. So I spent a lot of time thinking about this problem of the lack of independence in genomics, and actually it turns out a guy named Brent Pedersen in my lab, before he came to the lab, was working on the same problem, and wrote this paper with Subo Day about like how to actually handle this when you're testing for statistical significance of enrichment of overlaps between intervals. And so really what I want to show you is this scenario 3 here. This is defining, with these gray boxes, defining constraints on where you can shuffle things. So for instance, in our experiment, we could maybe refine our experiment by excluding centromeres, telomeres, and gaps, regions where we could never detect GWAS SNPs anyway, or never even assay them. So it's unfair to shuffle the SNPs to potentially those locations. And if it is unfair and we allow SNPs to be shuffled there, the significance that we observe from a Monte Carlo simulation will be much greater than it should be. Because if we basically define a neighborhood of the genome, or neighborhoods in the genome that are off limits, then that shrinks the genome, and it shrinks the space that we can shuffle to. So therefore the likelihood of random overlaps between SNPs and enhancers increases. Right? So the fact that we got 0 intersections between GWAS SNPs and enhancers is probably not entirely an artifact of the fact that we allowed those SNPs to be shuffled any old place in the genome, but maybe partially. So it might be that if we excluded telomeres and centromeres and gaps, then maybe we occasionally see a couple intersections, or maybe a couple hundred even. I don't know. We don't need to do the experiment. But I think it's worth thinking about, when you do these types of experiments, what are the boundaries? What are the constraints on the experiment you're doing? ChIP-seq probably has one thing for ChIP-seq that's often used, for instance, is ENCODE and other studies have defined regions of the genome that are well behaved, and therefore you can confidently call ChIP-seq peaks. So probably you want to limit, you might want to limit your shuffling to just those in-play regions of the genome where callers like MAX or other ChIP-seq callers are actually going to make a peak call. If you allow peaks to be shuffled outside of sort of the in-play part of the genome, you're artificially biasing or improving the significance of p-values from a Monte Carlo experiment. Everyone with me there? Okay, if there's no concerns about that conceptually, I just want to show how you do that. So with Bedtools, there's 2 different options: Include, INCL, and Exclude, EXCL. So here's a schematic of how that works from the Bedtools documentation. Here's 4 intervals in their native locations. If you shuffle them, they'll go to 4 random locations. Their size will remain the same. If you define an inclusion region, which is this green region, these intervals can only be shuffled to random locations within that subset of the genome defined by the include region. Okay? So that could be, for instance, the in-play regions for ChIP-seq experiments. The flip of that is if we use Exclude, this is a region in red that is off limits. You cannot shuffle intervals there. And you can use both include and exclude regions. So you might define this green box as an include, but as a separate file, you might have this exclude region. So therefore, the include region is subsetted to just this part that is not overlapped by the exclude region. Okay. what you define as your include or exclude regions really depends upon the experiment. But if you're going to do this type of analysis, this is something you need to think carefully about. What actually is in play with respect to the experimental design that generated the data that I'm analyzing. Okay. So that is the end of the Bedtools lecture. Hopefully everyone got the email that I've extended